Chapter 4, Section 4, Viable Counting of Microbial Cell Numbers. In Section 3, we did, this direct cell counting, using, hemocytometers and whatnot, those special etched slides. And I mentioned how the fact that when you look at them, you're just counting the number of cells there. You're not counting live cells. There's no way to differentiate the dead cells. So you could have an artificially distorted number. Here's all the cells there. Are they living, dead? Don't know. They're just there. Better method for counting to know what's there in a culture is what is referred to as viable counts. How many living cells are there? You do this by plating, taking aliquots of whatever it is, whatever broth or whatever, and plating out to see how many colonies per aliquot shows up. You can do this by two methods: the spread plate or what's called the pour plate. We're going to be doing the spread plate in lab in a few weeks. Pour plate method we're probably not going to do. It's half a dozen of one, 6 of one, half dozen of the other. I'm just picking one over the other because this one's just faster. Thing is, though, there are some parameters you have to operate under as you're doing these aliquots and you're making dilutions of said aliquots. The resulting plates, well, you're going to have to wait 24 hours to count them, okay? Because you got to give each of those one bacteria that's going to end up on the plate time to make 1 million clones. Those 1 million clones that you can see. That's one. Second thing you have to take into account here is that there is limits. You want to disregard any plates that you did dilutions of, of the original aliquot, and you end up with less than 30 colonies. Okay, that means you just did 2 dilute, and that could really distort any of your numbers if you try to count and it's less than 30. More than 300? Well, then the colonies are too close together and it makes it very, very difficult to distinguish one from another. So there's our parameters. Got to wait 24 hours and then count only the plates that have between 30 and 300. What you're counting are colonies, okay? The colonies, you will then, by knowing the colonies and the dilution, you'll be able to calculate what is referred to as the colony forming units. Basically, how many living cells, how many cells were alive in your original sample to give you colonies, 30 to 300 colonies per plate at this dilution. 1 to 100, 1 to 1,000, 1 to 10,000 dilution. The spread plate, you just literally take, and put certain volume of your bacterial dilution on a plate. You use a spreader, spread it around, incubate 12 to 24 hours, and then you come back and you count spots, the colonies. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. And then you figure out what the dilution was. Pour plate, well, you put down some agar. And once that solidifies, you come in and you pour the liquid on top. And you just let that swirl around. And it just becomes part of the cooling agar. And then, let that incubate overnight, 12 to 24 hours. And you come through, count, count, count. Why do I like the spread plate? Because I don't have to have extra liquid agar sitting around. That's it. I just have a bunch of agar plates. I put a, 1 ml of my culture or my diluted culture on them. Use a hockey stick, or a spreader, a glass spreader, and just smear it around. Done. We find that original samples, the tubes that you used, in lab to do your streak plates on. Could have anywhere from 1,000, 1,000,000, 1,000,000,000 viable cells per milliliter. Crap, that's a lot. It's a lot of cells any way you look at it. And that's a big range. 10 to the third, 10 to the sixth, 10 to the ninth. So what we commonly do is we take the unknown and then we make dilutions of it. We like to do tenfold dilutions. 1/10, 1/10, 1/10 dilutions. We call this serial dilutions or successive dilutions. How many dilutions? Well, do we think our original sample is going to have thousands, millions, billions, or trillions of living cells? So you take, and you're not taking all of your big sample. Here, this is 200 or 300 mLs. You're not taking all that. You're taking a small sample, 1 ml. You're going to take 1 ml of your sample, of your broth, your bacteria. You're going to put it into 9 mLs. Dilute it out, 1/10. You're going to take 1 from that and put it into 9. 1 from that into 9. 1 from that into 9, to 9, into 9. 1/10, 1/10, 1/10. So when you do the dilutions, this original first tube, tube 1, is 1/10 of the original. Tube 2, which is 1/10 of tube 1, which is, is 1/100th of the original. Tube 3, which is 1/10 of 2, which is 1/10 of 1, is 1,000th. 4, 10,000th, 100,000th, millionth. And then you take on 1 ml sample and you put that on the plate and you just smear it around with a spreader. Wait 12 to 24 hours and come back and count spots. The 1/10, the 100, yeah, you know what? That's way over 300. Too dense, can't count. Down here, the 6th tube, too few, too few, too few. Below 30, disregard those. But here, for tube 3, which is the 1,000th, there's 159 colonies. You go through and you count 159 spots. 159 of the 1 to 1,000 dilution means there's 159,000 viable bacterial cells per ml here. Or, microbiologist-wise, we like to number things logarithmically using scientific notation. It just makes things a lot easier for us. 159,000 becomes 1.59 times 10 to the 5th. That I can work with. I can, now know that the original here had 1.59 times 10 to the 5th viable cells per ml. Not total, per ml. So if this was 200, say 200 mLs total in this, 200 times that number is how many bacteria are there total. Anyway, do this. It's quick and easy. I mean, the hard part, it's drop, transfer, doing that serial dilution, transfer that to plate, smear, label, incubator, wait. And then come back, figure out which plates to count, and then you count them. This is how it's done in food, dairy, medical. The direct counting uses that hemocytometer. You don't know what's living, you don't know what's dead. This tells you how many living cells are present. It takes a little bit of time. They are now coming up with ways where you don't have to wait the 12 to 24 hours like we are, in our lab. They can do this within a few hours. So you'll see this being done in, food processing, dairy processing. Medical, they're doing this, taking swabs of different things, of instruments, of rooms and stuff, and then seeing if there's any type of growth, how much growth of what. So the problem becomes, like I said, this direct count of samples versus, what's on plates. The direct count tells you how many cells are there, just not how many were living. Okay, so that's a drawback for the direct counts. The drawback for the plating, whether it's the spread plate or the pour plate, what happens is the microbes that are present can't grow on that agar. Other ones can, but not all of them can. You're going to distort your numbers. You're going to skew your numbers one way. So when it comes down to it, there's good and bad. There's pros and cons for both direct or viable cell counts.